Assalamu alaikum students. Today our topic of discussion is ABO blood group system. So there are four types of blood groups: A, B, AB and O. Multiplicity of antigens in the blood cells, at least 30 commonly occurring antigens and 100 of other rare antigens, each of which can at times cause antigen-antibody reactions, have been found on the surface of the cell membranes of human blood cells. Most of the antigens are weak and therefore are of importance principally for studying the inheritance of genes to establish percentage. Two particular types of antigens are much more likely than the other to cause blood transfusion reactions. They are the ABO system of antigens and RH system. When blood transfusion from one person to another were first attempted, immediate or delayed agglutination and hemolysis of the red blood cells often occurred, resulting in typical transfusion reactions that frequently led to death. Soon it was discovered that the bloods of different people have different antigenic and immune properties. ABO blood types: A and B antigens, agglutinogens. Two antigens, type A and type B, occur on the surface of the red blood cells, also called agglutinogens, that cause most blood transfusion reactions. These agglutinogens, glycolipids and glycoproteins, are inherited. The locus for ABO alleles or genes responsible for synthesis of these antigens are on chromosome 9. There are three basic genes, A, B and H. Any two of these alleles are present to determine a blood group or genotype. Blood group refers to phenotype of these antigens. Blood types: multiple in blood cells at least occurring antigen which can cause antigen-antibody reactions are found on the surface of RBCs. ABO system of antigens and RH are important. So antigenicity leads to stimulation of immune system. So here you can see the four types of blood groups. This is the precursor structure of antigen. RBC having the structure of antigen includes glucose, galactose, N-acetyl glucosamine and galactose. While for formation of H antigen there is the presence of fucose which binds with galactose, and in case of A antigen fucose is also there, but here is another component, that is N-acetyl galactosamine, which is also bind with galactose. And in case of B antigen the galactose is bind with another galactose. So nature of A and B antigen: the type A and B antigens are actually complex oligosaccharides that differ in their terminal sugar. On red cells they are mostly glycosphingolipids, whereas in other tissues they are glycoproteins. An H gene codes for a fucose transferase that puts a fucose on the end of these glycolipids or glycoproteins, forming the H antigen or H substance that is usually present in individuals of all blood types. Individuals who are type A have a gene which codes for a transferase that catalyzes placement of a terminal N-acetyl galactosamine on the H antigen. You can see here the antigen part binds with the ceramide on the red blood cell surface. ABO blood group system. The various classical blood group system are due to presence of A or B antigen on the surface of RBCs. If only A is present, blood group type is A. If only B is present, blood group type is B. If both AB are present, blood group type is AB, and if both are absent, the blood group type is O. Corresponding to A and B antigens, anti-A and anti-B antibodies are present in the plasma, produced in response to exposure to respective antigen. So here is the Landsteiner's law, very important. If an agglutinogen is present in the RBCs of a person, then the corresponding agglutinin must be absent from his body. If an agglutinogen is not present in the RBCs of a person, then the corresponding agglutinin must be present in his body. We can explain this law by this diagram. You can see here the ABO blood group system, ABO and AB. So here is the antigen and antibodies. You can see here in when there is a blood group A, there is A antigen, but there is a B antibody. So its corresponding antibody is not present, which is the Landsteiner's law. So in case of a B blood group, there is B antigen and A antibody, while in O blood group there is no antigen, while both antibodies are present, while in AB both antigens are present and no antibody is present. You can again see the corresponding antigen and antibodies in respective blood groups. And their genotypes are also there. In case of O blood group, the genotype is double O, while in A blood group type, the genotype is AA or maybe OA, that is heterozygous or homozygous, while same is the case with B, and in the case of AB, the genotype is AB. Genetic determination of the agglutinogens. Two genes, one on each of two paired chromosomes, determine the ABO blood type, type O, type A, or type B. The type O gene is almost functionless, so that it causes no significant type O agglutinogen on the cells. Relative frequency of different blood types. O has a 47%, A 41%, B 9%, and AB has the least frequency, that is 3%. So maximum frequency is of O blood group. Subgroups of blood type A. Subgroups of blood type A have been described, the most important being A1, A2. However, the difference between A1 and A2 appear to be quantitative. Each A1 cell has about 10 lakh copies of the A antigen on its surface, and each A2 cell has about 2,50,000. The A2 RBCs can undergo agglutination by A1 antecedent. Antibodies against red cell agglutinogens are called agglutinins. Antigen very similar to A and B are common in intestinal bacteria and possibly in foods to which newborn individuals are exposed. Therefore, infants rapidly develop antibodies against the antigens not present in their own blood, on cells. Thus, type A individuals develop anti-B antibodies, while type B individuals develop anti-A antibodies, and type O individuals develop both, and type AB develop neither. Most of them are IgM and IgG immunoglobulins produced by the bone marrow and the lymph nodes. Immediately after birth, the quantity of agglutinins in the plasma is almost zero. 2 to 8 months after birth, and an infant begin to produce agglutinins, anti-A agglutinins. When type A agglutinogen are present in the cells, a maximum titer is usually reached at 8 to 10 years of age, and this gradually declines throughout the remaining years of life. You can see here the titer of agglutinin is maximum in about 8 to 10 years of age. Then with the increasing age, this titer is decreasing. This is the normal blood smear under microscope, while agglutination under microscope. You can see here, there is the clumping or agglutination of RBCs. You can also see here this one, this, there is clumping or agglutination of RBCs. Secretors and non-secretors. These antigens are found in many tissues, glycoproteins in addition to blood. This includes salivary glands, saliva, pancreas, kidneys, liver, lungs, testes, semen, and amniotic fluid. A dominant gene, that is SC, specific enzyme, genotype SE over SE, and SE over SE, that is homozygous dominant or heterozygous dominant, will be secretors. The soluble blood group antigens block the binding of microorganisms, while non-secretors susceptible to infections like Candida albicans, that is the fungal infection, Streptococcus pneumoniae, Haemophilus influenza. So the secretors have a benefit over non-secretors, as they block the antigen by binding the antibodies present in their secretions. This is the inheritance pattern of ABO blood grouping. For blood types, their genotype, and you can see here in A blood group, the genotype may be AO or AA, and same is the case with B blood group. Blood group typing and matching, and there is a cross matching as well. So blood group typing. Before giving transfusion to a person, it is necessary to determine the blood type of the recipient blood and the blood type of the donor blood, so that the bloods can be appropriately matched. The red blood cells are first separated from the plasma and diluted with saline. One portion is then mixed with the anti-A agglutinin and another portion with anti-B agglutinin. After several minutes, the mixtures are observed under microscope. If the red blood cells have become clumped, that is agglutinated, one knows that an antigen-antibody reaction has resulted. Blood agglutination by antibodies, ABO donor-recipient compatibility. Add a drop of anti-A antibody on the left side to blood drop A, while add a drop of anti-B antibody on right to the blood drop B. See if drop A and drop B are clumped by antibodies. And this is the case of type O blood group. Here there is no clumping, so in drops A or B, that's why this is type O blood group. And in case of type A blood group, clumping is in A. and in case of type B blood group, clumping is in B, and while in AB blood group, clumping is in A and B blood drops. So you can see here the large protein found in the plasma protein, that is antibody with two combining sites. Anti-A antibody attaches to A antigen, while anti-B antibody attaches to B antigen, and anti-RH antibody attaches to RH antigen. So you can see here antibodies attached to antigen on the surface of RBC membrane. You can see these all the three antibodies are attached with the RBC. So it is the blood group having all the three types of antigens, as well as they are all these three types of antibodies are there. So this is AB positive blood group. So different blood types with the anti-A and anti-B agglutinins in the sera. You can see here in the O there is no antibodies, while in AB both antibodies are there, while in A you can see the anti-A, that sera is anti-A, while in B there is anti-B sera. So the blood typing, uh, you can again see in case of there is no reaction of anti-A or anti-B antibodies in case of O blood group, while the anti-A antibody react with A antigen in A blood group, and B antibody reacts with B blood group, because this B blood group has B antigen, while in AB both antibodies are present, both so both antigens are present, so these both antibodies can react with both antigen. So which blood group? Guess. You can see there is a clumping in anti-A part, and no clumping in anti-B or anti-D. So it is your A negative blood group, and you can compare it with control. So clumping is only in A, because there is A antigen is there, so this anti-A antibody combines or agglutinate with the antigen. Donor's red blood cells are mixed with the recipient plasma. The reaction between the donor's plasma and recipient red blood cells is ignored. Why? Donor's agglutinins in recipient blood get diluted and disintegrate after some times. That's why during the reaction between donor plasma and recipient uh, red blood cells is ignored. So in cross matching, the importance is given to donor's red blood cells and recipient's plasma. You can again see the O blood group, that is a universal donor, while so there is no reaction is there, while the AB blood group is a universal recipient. So no reaction on the AB side. You can see here there is no reaction, while in O also no reaction because it is universal donor, and AB is universal recipient. You can also see here when red blood cells compatibility table. So O recipient and O donor, there is no reaction, but when there is O negative with O positive, there is a reaction. The transfusion reaction is there. So other tests before blood transfusion on donor blood for malarial parasite, for hepatitis and AIDS. And clinical significance, blood transfusion, certain blood diseases, to detect certain blood diseases, and to resolve the disputed paternity, as and also in the forensic medicine to detect the criminals. So you can see here there are two types of grouping, that is ABO cell grouping and ABO serum grouping. So here you can see the recipient has a blood specimen from where the recipient RBCs are there, and in case of you can see when there is a recipient blood specimen, there is the extraction of only recipient serum in ABO serum grouping, and in recipient RBCs, then there is an introduction of antibody A. So recipients RBCs, and there is an introduction of A antibody. So if there is agglutination, means the A antigen is present on the RBCs, because for A antigen there the corresponding antibody is A. So if no agglutination, means the A antigen is not present on the RBCs. So when there is a recipient serum and we add the group A red cells, then again two results. If there is agglutination, it means anti-A antibody is present in the recipient serum, because the A red blood cells correspond or form a transfusion reaction with the respective antibody. If no agglutination is there, it means anti-A antibody is not present in the recipient serum. So it is all about the blood groups. If you have any confusion regarding blood groups, you can contact me. Thank you.